Okay, so we're going to start the second part of this, I guess, session on sustainable chemistry, efficient manufacturing, and green chemistry. And we have two more talks. And this one is from Dr. Eric Fang. Got your PhD in Toronto and a postdoc with the famous Eric Jacobsen. And then joined Amgen, process chemistry, and then you founded Snapdragon with a couple of other people. Yep. And Snapdragon is part now of Cambrex. And you're going to talk about the flow chemistry into liquid phase peptide synthesis and an overview of what you guys are doing to make the things more efficient, right? Yeah. Thank you.
Thank you. You guys can hear me. So I guess most of people in this peptide or oligo world probably doesn't know, most people don't know Snapdragon or Cambrex in that sense, even though Cambrex is probably one of the largest CDMOs in the industry of making APIs. So why we're here, right? Maybe we're in the, as some earlier speaker says, we're in a very exciting time of a gold rush. But I think, this is there's probably some little bit more nuances to that. So hopefully tell you a little bit story about how we apply the flow chemistry. I think in some ways, I think it's really some inspiration or some tools we developed during the flow chemistry that can readily apply to developing liquid peptide synthesis technology. Because as we review these issues, a lot of times manufacturing is one thing, but actually developing those fragment-based liquid peptide synthesis is actually quite challenging and very time-consuming. And that's what we view is that why this is not applied more widely. So give you a quick sense, don't know Snapdragon, probably we're a very young company. So we started this flow chemistry technology and services about 10 years ago. So our focus was really try to use the flow chemistry to solve some challenging, typical small molecule processes. Really enable those processes, applying photochemistry, applying fluoridation chemistry, some of the known challenging chemistries. While we're doing this type of development, one of the things which is not appreciated at times is that some of the associated knowledge or technologies that has to go into it. Those including software, hardware, and in some cases also opportunity. When you are in full control of automation software, really the opportunity is really at your fingertip. So we can apply automation to antenna experimentations. We can do algorithm-driven optimizations, and many, many things. So those tools actually become very handy when we are applying those to, I would call, precision chemistry. In some cases, I think peptide or oligonucleotide requires far more stringent controls from engineering and automation and precision standpoint that is really requires those tools. And that's actually not very well readily available in traditional batch chemistries. So we're applying those tools to a rapid scale up to enable some clinical stage deliveries. And we also applied those tools to GMP manufacturing of small volume APIs and many things. Because of those kind of complexities. So we have pretty broad coverage in terms of development and commercial, clinical stage services in flow chemistry, peptide, polymeric, APIs, etc. So it really takes quite a bit of a skill set to serve this type of services. So I think one of the things we did right at the very beginning, foundation of the company, is to really put a highly multidisciplinary team together. So really involving software engineering, automation engineering, and mechanical engineering, as well as chemistry, analytical, and other traditional disciplines to enable these things. And the software and data really is in the center of this. We have, over the years, built many different tools and made many deliveries of critical deliveries to our clients for clinical stage APIs. Some of them are very critical intermediates. So, we have had the ability to access far wider process parameters, minus 70 or below or 300 above. These all become accessible when you are running flow chemistry, of course. Things like continuous crystallization or packback technology is also a very, very good technology to intensify processes or enable some processes. One of the examples I want to present is flow chemistry here, which is probably not as familiar in this audience, but I want to kind of put it as an example that in some of the cases, really precision chemistry is something that required to scale up, and technology is pretty available in this age. And of course, execution application is very important. So this is pretty simple chemistry in some sense, but it's also pretty complicated because it's a very complicated molecule. So methylating a tertiary alcohol, but the problem is when you do deprotonation, it has a very short lifetime of this intermediate. So, and very rapidly loses its activity. Then when you get hit it too hard in the downstream methylation chemistry side, basically you generate a bunch of impurities you cannot purge downstream. And this is the API step. And we designed a continuous flow process, and we can precisely to deprotonate this in short 15 seconds. Then we apply 2 stages of CSTR, again, very precise, to get the methylation complete, and then quench immediately, and then
CSTR, can you say?
Yeah, it's a CSTR is the term called continuous stir tank reactors. They look like a basically reactor, batch reactor, but have material flows in and flow out. But they actually one of the flow reactor systems. So you can model this type of chemistry when you understand reaction kinetics, which is something that we have developed quite a bit of a skill set on. And you can model that, and you can model at different scales and actually getting results spot on in many cases. So it's a type of activities you can do based on very simple chemical engineering. And based on those, you can design standalone equipment to serve those purposes. And this is one of the equipment that we actually delivered to our CDMO, third-party CDMO partner that applied for this GMP production. So you can see that through very small scale, the reason we actually intentionally slowed down the development because the material availability was pretty tight. And with about 6 months of cycle time, we're able to turn a proof of concept stage of a couple gram reactions to tens of kg productions on the GMP at another third party CDMO. So those are the type of muscles we actually have built while we're developing our own technology as well as understanding critical chemistries and apply to it. And that's the type of tool, I think, when we kind of look into this opportunity for peptide, and it really is one of the fundamental problem is, as I think I don't need to do any further introduction regarding this criticality of peptide scale up. I think all the speakers here has talked about this, and I can quickly skip those slides, even though I have prepared some of them. So this is, well, what is the fundamental problems, right? Fundamental problems that our bodies are organisms really fast making peptide bonds, and we're basically a couple order of magnitude off, and the precision, everything is so good. Now, of course, those are applied to those coded amino acids, right? And that's one. Second thing is that they're generally relatively large. If they're too small, it's pretty easy to get those peptides presented to the organism to break down pretty quickly. So you have to engineer those pretty specifically for that. Of course, chemists, I think a lot of people here has working on this for probably many decades as well. This is many generations of chemists working on this field, and I think we're getting approaching to the sort of efficiency, but still we have good amount of error rate regarding each coupling step. The speed is still not there. You talk about each amino acid, 2 order of magnitude probably speed-wide compared to. But I think that really the fundamental problem is the engineering part, right? So the engineering part is that you have to basically keep repeating the same thing and get rid of the impurities and all that stuff, which I don't need to present to this audience on that side. For solution phase chemistry, we are talking about this one, 60 years late, right? Later. This is actually one of those confirmed. This is a New York Times I actually put out from the archives. In 1966, which is the same, another side, it was the Red Guards and guarding the Peking, parading, all that stuff. This is actually the synthesized insulin in solution phase 50 years ago, 70, that's 60, 70 years ago, 60 years ago. Okay, so that's been done. Yield was somewhere around 1 to 3%. I think it was confirmed by some German scientists that way. But if you really look at this problem, right, it's a heroic effort, and I think there's a published work later you follow up on that side, and the material actually crystallized out, and it was all kind of rigorous studies that confirms it. But this is just simply too expensive to commercialize. Just that's the bottom line. I'm not talking about something groundbreaking anything. Just really just about a cost. And now we see that even at this age. The trick is, I think, when a Nobel Prize is just put stuff on a solid, right? If you look at this one, it's pretty interesting that's a very simple trick that the chemists apply to it. Meanwhile, I think it was also technology similar times, so-called recombinant technologies, all competed the solid phase for many years. I think there's probably some few reasons why that has been dominant, right? SPPS been dominant for many years. I think one thing, obviously, is that it's still pretty expensive to produce. And is anything that large enough to be similar as the sort of a innate type of insulin or some sort of molecules, recombinant technology is far cheaper to produce and pretty reliable to do that. But I think what's interesting here is that the discovery chemistry has delayed, and I think a recent discovery regarding some tricks to play, right, extend lifetime of these therapeutic peptides and through non-coded amino acids, so on and so forth. Really, I think is really most exciting part, and those ones has to be synthetically made that way. I think this is, again, I don't have to go through this one, but it's pretty standard process of solid phase peptide synthesis. I think I've done one. Pretty quickly skip that. And this some of the analogies making, some of them probably can agree with me, but some of you may not. But I think the analogy is that, I think this is we all in this audience all agree that cost and sustainability is one of the key things, and fundamentally was driven by some of the key challenges of chemistry and physics, right? Those resins once grow large enough, it looks like a gel, and it's very soft gel. I think we have those in our laboratory as well. I mean, it takes a tremendous amount of volumes, and the volume efficiency is not very high. And I think before 2020, very few molecules made large enough to be interesting. And I think I was fortunate enough to actually worked on Amgen's valcosatide, and we saw at time we called AMG 416. And I actually interesting enough to did some work on this project, and I really understand some key challenges on that. So this is really, a lot of times in the peptide space, people don't actually appreciate as much of those sort of reaction kinetics, and this is difficult to study. And this is a published paper we did about, work is done about 10 years ago, more than 10 years ago now. But if you look at the solid phase, I think, if you look at reactions, it behaves like first order reaction kinetics. But at the same time, amino acids once get activated, is degrading as well, and especially on this arginine chemistry side. And you can kind of model that, and really last bit of reaction takes forever to complete. I think we've seen that. And of course, this is actually a pretty small peptide. This is only AMERS 416, and you're talking about much larger peptide. You start seeing aggregation on solid phase, and the reaction even longer. So intensification in some ways is really limited by the fundamental physics or reaction kinetics. And again, I'm going to skip this slide because all those things has been talked about. And this is again recycled with another side of both Frederick and other speakers that talked about this. I think it really divide and conquer, right? And I think if you look at this one, all the fragments are still made in solid phase peptide synthesis technology, and it still has some fundamental issues of the PMI that contribute to this. Although this one maybe it's somebody else's dirty job, but it's not visible to the end users in some cases. So what we're interested to see is how can we turn this into a total solution phase peptide synthesis. So really take advantage of all the work both Lilly and Roche has developed in the downstream late phase convergent synthesis, but all the fragments need to be also made in solution phase, right? And the way, the way we view is that we're taking on this tag-based, tag-assisted solution phase peptide synthesis to make the assemblies. And I think we're developing the second gen type of processes. And the advantages of those is obvious. Quality all have been touched by some of the previous speakers: quality improvement, parallel quality risk management, supply chain, etc. And we have been a part of Cambrex, which I didn't touch as much, that we were acquired. Even though we were 10 years old company, we were acquired by Cambrex, and Cambrex is a company that has all the way traced back to Oxford Nobel's time. That one site in Karlskoga actually utilized the Nobel's. So it's 100-something years old company in some ways. So we have all the big reactors, and I think portability part is a lot of companies looking for as we scaling the peptide synthesis, and then take advantage of the scalability in those very large reactors. You're talking about 16,000 liter reactors, and really provide the economy of scale. So I think this is the industry demands it. And I think a lot of chemistry side or development side really need to kind of fit into that. So really, what we're looking at for the second gen fragment synthesis using solution phase is really solving some challenges regarding tag optimization, reduce the cost, and really understanding some of the key chemistry parts. Some key considerations we're doing this, obviously, is on the, when we're chopping a big peptide into smaller fragments, where to do, right? And obviously, some solubility is something key to consider. Downstream coupling efficiency has to also be. And tag side, optimize the tag that instead of an equivalent of 1,000 molecular weight or Dalton, we don't really need that big when we are talking about just want to make 10 mers or 12 mers in that way. So we actually can use very lean tag to assist it to solubilize. And also we can open up a space other than FMOC. We actually can start using CBZ technology. So regarding what type of tag selection, so I think this is really playing into the organic chemistry. So it's just orthogonal protecting group strategies that has to be able to survive. Some that has to be cleaved off, while some of the so-called soft cleavage, and some of the protecting groups on the side chain has to be stay on. So there's a lot of interesting things that you have to consider, and I think by the end of the day, it's a simple organic chemistry with orthogonal protecting groups, which you're kind of learning that, through sophomore organic chemistries. So one of the examples I want to share here is, this is a 11-mer, give or take. When we're in the first iteration, we used LQ as the C-terminus ester. And we will try many different optimization conditions, try to cleave off and through saponification, other means, and really have a pretty significant amount of epimerization as well as side chain deprotection of those tubular esters. Well, we switched it to the allo-based tag, which is very easily cleaved by Palladium chemistry. It's completely orthogonal to all the protecting groups around this molecule. You can see very clean cleavage without any epimerization. So there's a lot of things you can play with while you're doing that. And then FMOC, this is basically the biggest player. I think people almost forgot there's a tubular, the bulk amino acids or CBZ amino acids. I think those become almost irrelevant when you're starting predominantly using the FMOC for SPPS. But there's a lot of advantages of using CBZ technology. One of them is that the cleavage conditions are super mild. And it's a complete neutral in a way. So you can, a lot of that early couplings that has issues with diketopyridine formations actually completely sort of avoided when you use CBZ technology. So that's something that we use quite a lot. And obviously, when you use CBZ, you don't generate those impurities. And the only byproducts toluene, CO2, and water. So right away, you can go to the next step coupling. So now sometimes that CBZ don't survive very well, or some of the conditions of the side chain protecting groups don't survive very well with CBZ conditions, then we still can introduce FMOC in the process. And example here I share here is the tryptophan can be hydrogenated and create impurities, but by switching over to FMOC, you can avoid that. Now, the next thing is the fragment selection. I think those are actually, during the development, SPPS can help quite a lot, which we use quite extensively. So really deciding where the breakage point, and that's a retrosynthetic analysis point. Once you actually have a proof of concept, then you can focus on the development of the liquid peptide synthesis using tag-based solution phase chemistry. So protecting groups, we can put a hybrid approach. Some of them actually have FMOC, some of them CBZ technology. So this is a AMER we made, has triple protecting group, has FMOC chemistry, and mostly actually using CBZ technology. And I think this again, it's pretty simple chemistry. You know, the product stays in organic phase, and then all the washes is using aqueous to get rid of the coupling reagent byproduct or from the FMOC washes. So one of the tools we actually have developed doing flow chemistry side is so-called auto optimization tool. So it's a black box tool. You can view it as a DOE on steroid, but it's just automated, and it's just making its own self decisions how you traverse to the global minimum. So this is a dinucleic acid chemistry we optimize. It's not very intuitive in terms of what type of knobs you can tune to optimize this chemistry. But we developed this online HPLC system, which is very similar as a LIDI system, but we took advantage of more commercial tools and linked the easy sampler, mallow-Toledo easy sampler with the UPLC with our SIS sample box, and then we actually parameters, variables on our LabOS software control, and we apply this LabOS, and we dramatically increase the optimization efficiency. And you can see from this pack bag hydrogenation of this CBZ protected amino acids, and we put there after very quick 16 experiment run, we can optimize this chemistry very, very high efficiency. And the process can also be run in batch, so it's pretty portable whether you're, from the manufacturing standpoint. So you can run this chemistry even on the very slight pressure of the hydrogen, and that takes off pretty quick. And we scale this one to 10 kg scales now. And that's the sort of efficiencies you can see that, we in our kilo lab, we really have a very small footprint kilo lab in our facility in Waltham, Massachusetts. And we can produce this 10 kg scales for this. And I think a lot of things we do, just a small amount of amino acid excess you need, and primarily use methyl THF as a solvent that retains the product. And it's not actually, it's just recycling in a way. And I think this is one of the key many slides, I would say. In this case, we're looking at a PMI of 350 for AMER, and that's average 44 amino acids. I think it compares very favorably for the others, LPPS or SPPS. And if you look at the waste coming out of, majority of the waste is aqueous washes, and I think some parts DMF and methyl THF is another part of that waste. Now, when you're looking at this, again, I think you go back to the original question, why people not using that technology, even though Roche has actually developed first approach to this about 20 years ago now. And I think it really comes down to sort of the how easy you can get addicted to SPPS, because a lot of that technology is fully automated, and you can get the 40-mer very quick, while you probably need to take a couple of months to actually make a 40-mer when you do LPPS in that way. So I think that's really one of the key challenges. I think certainly we want to accelerate this development, and I think I should apply more, especially to the potential high volume liquid peptide synthesis. I think the opportunity is there. And the money question is that how can you automate, right, the LPPS, and how can you do just as easy kind of automation as a SPPS? I think one of the things you appreciate that, I was jokingly saying that one simple trick that won Nobel Prize, but I think it really is a fundamentally important trick, because filtration is all you need right, to retain the stuff, and everything else go to solution phase. Now we are talking about 2 layers. Both layers are liquid. So how can we do these automations? And I think we are working on it, and we're applying all kinds of different tools and automation, PLCs, and even AIs in this case, because we're using computer vision to help us to ensure, have some sanity checks while the sensors fail. So this is actually a real run. I'll just probably show a snippet of it. So this is a 3-tank system. Is designed for both doing FMOC chemistry as well as doing the CBZ chemistry. Okay, so the second tank is basically a palladium on aluminum that we can charge in. And then the first front tank is actually just activation of the amino acids. So if you look at this, and I'll show probably a few seconds of it, and this is actually where some wash is happening, after prior couplings complete, and you basically you see the color change going on, the reaction stirring, coupling going on, and you will very quickly see some washes going on. The drop, wash, drop, wash, and nice and clean, right? And now we'll see the front end tank's going to start activation of the second amino acid. And basically, there's a lot of automation ongoing there. And I'll just try to see what I can say. Okay, there we go. So basically, you're going to start seeing the activation coming, right? Then dropped into the second tank, then reaction coupling going on, and then so on and so forth. You keep going on that side. So for this time, I don't have that. So anyway, so I think hopefully you're going to see some interesting things from the talk, and the way we're thinking about this, sort of a holistic way of thinking about a technology application to really fundamental challenging problem as well exciting problem. I really want to kind of acknowledge my team as really talented team, and with a very large number of people involved, and obviously a lot of great customers as well, and give us difficult problems to solve, and it's just very privileged to be at Snapdragon and working with customers. And it's a quite exciting young crew with some good experience behind as well. So thank you for your attention, and I'm here for questions.
Anybody has any question?
So the chemistry is very nice. Congratulations. There's one thing that I would like to ask you regarding the large-scale commercialization of this. And one of the advantages in this moment of FMOC chemistry is that the amino acids are damn cheap. On the other hand, I think your CBZ amino acid, because they don't have this huge demand out there, they are still quite expensive. Does Cambrex produce in-house, and can the customer of Cambrex take advantage of this integration in-house? Do you still source outside? How is your plan for the amino acid?
It's a good question. So I think this is always the question of demand, right? Economy of scale. I think that's we all have to appreciate that part. Yes, we actually have a pretty good supply chain to supply those things. Those are all well known. And if you look at, and then there's a lot of the amino acids still produced in China. I think we have a quota on those. And at the largest scale, we're talking about metric ton scale. They're cheaper than FMOC chemistry, because that's a fundamental mole per mole. If one thing you look at the kg, size. Another thing you look at mole per mole base, right? And they're very cost competitive, or even cheaper on that side, because fundamentally it's just find somebody provider with that process, and they can make it for you.
Any other question from the chemist here? Well, I think, maybe one question for you.
Of course.
So what have you done at, let's say, large scale, I would say like 100 kilo scale with your CBZ? Have you done 100 kilo? And then what will be the size, like 8 amino acid, 10,
Yeah.
4?
This is actually we're working with the customer to move it towards that direction. So I think this is a stepwise process because this is pretty new technology, and I think a lot of that, is moving that scale. So currently, we have a 11-mer that's being actually pretty well tested on the kg scale, and then ready to basically process characterization and move on to 100 scale, hundreds of kg scale, very large reactors in our site in Europe, in Italy actually. So it's a work in progress.
Okay.
I have one real quick. At the standpoint of the CBZ groups, one of the things that often comes up with the C groups is the thiol poisoning with the deprotections. How have you dealt with that with your technology?
So right. So I think that's those cases. First, we test if it is a case on that side. Often it is a case. FMOC is just rarely usable for our technology. So basically, you saw that similar cases that we replaced, for example, tryptophan chemistry with FMOC chemistry. And in fact, a lot of times we actually kind of got into a lot of FMOC chemistries in this liquid peptide synthesis, which I don't have time to dig any deeper, but it works just as well as with FMOC.
All right. Well, looks like we're running out of time.
Okay.
So if you have a question, you can find Eric in the next few days. And we're going to...
Thank you very much again.
Thank you. Thank you.